mycota. Okay, so tomorrow it will be lab exercise 8 and 9, both of them together. I am going to dispense right now with dissections. Okay, what we'll do is, because I just want to go through the diversity, okay, for it all the way up to exercise 10. Monday will be exercise 10, and then on Wednesday will be the lab exam. Okay, and so a little bit later I'll talk about the lab exam. I've already done some talking about it, but I'll do a little bit more. So after spring break, come back and we will do the dissections, okay? Because there'll be an earthworm dissection, there'll be a cockroach dissection, frog, and a shark, okay? So, and then later on, a fetal pig. Okay, the fetal pig will be a very detailed dissection that will take 3 lab periods, okay? And it's going to be preserved. It's going to be a preserved pig. The cockroach, earthworm, frog, those will be newly killed animals so that they're going to be very fresh without preservative.
For lab 8 and 9, would the lab quiz be everything on lab 8 and 9, or just
What's that?
the lab quiz is going to cover both 8 and 9 as well?
Yes. Yeah, Wednesday will be 8. 8 is real short, and 9 is a little bit longer. And so the pre-lab quiz, of course, they're separate pre-labs, but nonetheless, it'll still be only a 4-point quiz. Okay, no more than 4 questions. You know, so that's the way it'll be. Okay. Yes. Say what?
Oh my God.
I still didn't understand.
He still doesn't know.
What's the approximate estimated test grade?
I tried to get them back in the week. But you notice there was very little multiple choice. Okay, so that means might be a little bit more, but you'll get them before spring break. Yeah, I tried to get exams and quizzes back very quickly. All right, so basidial mycota. This is the last, and I already went over it. That basidial carp clamp connections. So I went over the apomorphies there and so forth. These are some examples. We're mostly concentrating on gill mushrooms and polypores. Actually, that's not a polypore. This is a polypore, but nonetheless, some of the details are different depending on what you're talking about. All right, so this is a gill mushroom. This is the life cycle. Now, one of the things about the life cycle of the basidial mycota, you notice here that it doesn't show an asexual stage. There are some basidial mycotes that do have an asexual stage, and when that's the case, it's canidia, but there's very, very few that do this. This is one group that is primarily sexually reproducing. So let's just start right here. Okay, we have spores. Okay, and of course the spores are haploid, and when they germinate, they will grow into a haploid plus, haploid minus strain. Okay, once again, they have to be different mating strains, plus and minus. These are very short-lived, this haploid mycelium. It only lasts until plasmogamy, and then that's the end of it, okay? Unlike in the other groups. So we get plasmogamy, that produces a dikaryotic cell, and which will grow into an extensive, long-lived dikaryotic mycelium. The original plus and minus haploid mycelia die off, okay? So that the major fungus that exists here underground is dikaryotic, okay? And when it sends up fruiting bodies, fruiting body is called a basidial carp, okay? And so every cell there happens to be dikaryotic. Where the reproductive parts are under the cap of the mushroom. Okay, this mushroom is called basidial carp, but it's on these gills. Okay, and the special types of reproductive cells are called basidia. Okay, basidium singular, basidia plural. Okay, and so what happened? You lost me. again. I'm not there. Huh. Okay, we're back. All right, so, and all of the basidia are just on the outside of the gills. Now that particular layer that has the basidia is called the hymeneum. That's just like in Ascomycota. The hymeneum in Ascomycota was the layer of asci in the cup, and here it is the layer of basidia on the outside of the gills.
So the basidia are the spore-producing cells, or are they the
The
whole?
basidia is the, okay, the basidia is a cell, and that is one where meiosis will take place and spore production will be. The layer of all basidia is called the hymeneum. Okay, yeah.
And this
All
will
right.
be under the cap, right? Under
What's
the
that?
cap. This will be under the cap of the mushroom.
Yeah, yeah, so it's on the gills, and I'll show pictures of it here in a moment. So, in fact, let's see if I can back up. Yeah, so these are the gills, of course, and when you zoom in on there, this is a longitudinal section, longitudinal section through the gill, and these are the basidium. That's a basidium, that's a basidium, that's a basidium, that's a basidium, and those are the spores, okay? This here is sterile hyphae, sterile, non-reproductive hyphae. Now, what is taking place here is, first of all, so I'm going to exaggerate this, have a basidium. First of all, of course, it is dikaryotic. We get plasmogamy. So I'm showing the progression as it develops. Okay, this is all in the same cell. We get plasmogamy, which produces the zygote nucleus. That will immediately undergo meiosis, so that we now have 4 haploid nuclei. These nuclei will migrate out to the end of the basidium and mature as on stalks as basidial spores. And there will be 4 of them, typically. Okay, so these are called basidial spores. Okay, and of course they are haploid.
What the basidial spores, the red part we refer to as
Yep.
the last cap?
Yes, they are. So these right here are the basidial spores. Okay? And the basidium is now empty, because all the cytoplasm has migrated into the spores themselves. And then these are released, and we're back at the beginning of the life cycle. Okay? And so that completes it. All right, so that's the last group. The story is pretty much the same as in Ascomycota, just only the words are different. Want to remind you, myospores, broader concept, okay? Broader concept. In the Ascomycota, the myospores are called ascospores. In the Basidiomycota, the myospores are called basidiospores. Okay? That's all to it. All right, so just to finish up here on the fungi, just a little bit of aspects of them. Oh, and this here is also a Basidiomycota, but instead of having, this is called a polypore, instead of having gills, it has these cylinder-like structures that go into the cap. Okay, and the basidia and the hymenum line the interface, the inside of that cylinder. And the basidia, same thing will happen there as happens here. Okay, and I showed this before. This is called a fairy ring, and that's not a fairy. Okay, this is a fairy ring, and it's usually these mushrooms come up at the very edge of the mycelium that's underground. And so that mycelium, these are all the mushrooms coming up from one individual mycelium, okay? And it's probably associated with the roots of this tree. And so you frequently will find this type of arrangement with these critters. Okay, now as far as their importance to humans, applications here, just some terms that are not phylogenetic, they don't mean anything except they describe just only structure. What you usually call a mold is an asexually reproducing fungus, okay? So that's real common in Zygomycota. There are a few molds that are also in the Ascomycota. For example, Penicillium is a mold, Aspergillus is a mold, and some fungi are, okay, in fact, Penicillium said it's a mold. It has no sexual stages, but it is a source of penicillin, antibiotic. That's where the first antibiotic was ever discovered, was produced by this particular fungus. Good Lord, you guys like blue cheese? All that green or blue stuff that's in there, that's fungus that you're eating, okay? And so they intentionally will inoculate the cheese during the production here to have the fungus grow there, because the fungus is taking up, digesting some of that organic material and putting out waste products, and people who like this blue cheese sort of like the taste of that waste product, okay? So that's where it comes from. Yeast, this is a unicellular fungus, okay? And it doesn't have any phylogenetic or taxonomic meaning, because all throughout the fungi we have few species here and there that are single cell, okay? So Saccharomyces cerevisiae, this is the most important one. Let me see, there it is. Where is it? I guess I don't, yeah, there it is, Saccharomyces cerevisiae. This is the type of fungus that you use, yeast that you use to make your bread. It's also used in the brewing industry. It's also a model organism. Okay, now what does it mean to be a model organism? A model organism, you know, we have 1.5 million described species of things, from bacteria all the way to eukaryotes. And it is impossible to study them all in detail. And so what a model organism is, are certain groups, certain species within a taxonomic group that have been studied intensely. Okay, so for fungi, this is a model organism, yeast. So we know a lot about its genetics and so forth. Other types of model organisms include the mouse, okay, and rat, because they're used extensively in research. Drosophila melanogaster, that's another model organism, and so forth. Okay, and among bacteria, Escherichia coli, that is a model organism. These are organisms that have been studied very, very intensely, and they tell us a lot. So this is still a yeast. Other types of yeast, Rhodotorula, this usually is found on plastic. There is a yeast, it's sort of slippery, so it's on shower curtains quite a bit, because when you take a shower, you're throwing off not only soap, but also skin cells, and that gets on the curtain, and that's what they're feeding on, okay? Candida, that's a genus that causes a yeast that can cause not only vaginal infections, it's very common for, in human vagina, but also in urinary tract, it can have infections there as well. So these are sort of bad yeasts. Okay, so this is very, very important. I guess I had that model organism up there. Some of fungi for food, like in the stores, agaricus. That's and now there's even other types of genera of mushrooms that are used for food as well. Truffles, but there's also some bad ones here. Parasitic, like this is a fungus that grows on skin. So aphids foot, jock itch, ringworm. In fact, these two cause the jock itch, aphids foot, same species. It's just growing in different places. It's given a different name depending on where it happens to be growing. By the time you reach the age of 30 or so, probably most of your toenails are infected with some sort of fungus that is growing inside of the toenails. That's real common too, hard to get rid of. As far as their ecological relationships that are important, is they're one of the major, fungi are one of the major decomposers, in addition to bacteria, okay? They're breaking down organic material, and in fact, this is one of the rare groups that can break down this polymer that is found in wood, called lignin. And only fungi have the types of enzymes that can hydrolyze and use the components of lignin. So this is very, otherwise, if it weren't for these decomposers, we would have leaf litter and branches that would just build up on the forest floor, and you wouldn't get any recycling of the nutrients. Okay, they'd be locked into complex structure. Now, these decomposers, however, they just recognize wood, and it doesn't matter if it's wood that's on the forest floor or if it's wood in your home. And so sometimes when your wood in the building gets wet, then that provides a place for these fungi to cause rot, wood rot. Yes.
So the trichomonas not able to
Trichomonas, yeah, they can, yeah, but they're so small that, you know, so the termites are very important ecologically too. They're also decomposers. Yeah, so that they would fit in it, but it's the microorganisms doing it, not the termites themselves. Right, but still, the number of species of fungi that are decomposers far outnumber the number of species of termites. Yeah. Okay. Furthermore, certain toxins, aflatoxins. In fact, the fungi, they are biochemical specialists. They make all sorts of what are called secondary metabolites. Secondary metabolites, some of which we don't really understand what they're for. A lot of these are defensive to protect our own food supply. Some of these toxins, they prevent other species of fungi from invading the food supply. Okay? Aflatoxins. This is usually, how many of you have peanut butter in your house that's over 8 months old? You're still eating it. You're probably getting a lot of that stuff, okay? Even though you don't see the fungi, it's there, and they're producing, usually peanut butter accumulates a lot of aflatoxins as time goes on. These can be very damaging to the liver, okay, in the long term. Some of these are pathogens. They cause disease. For example, Dutch elm disease. This was a fungus that was imported, I think, from Europe, because back in the 1950s, 40s or so, a lot of American cities in the United States, they planted Dutch elms. Okay, they were everywhere. But as this fungus came along, it spread all through the cities and just completely devastated Dutch elm trees. We don't see too many of those trees anymore. American chestnut. This was a major tree in the eastern forest. Right now, if you drive over into the eastern forest on the east side of the United States, the typical forests there are dominated by oak and hickory, various species of oak and hickory. But when Europeans first came to this continent, this was a major tree in the eastern forest. It was one of the dominant trees there, but you rarely find it now. There's a few groves that are highly protected, but this was attacked by a fungus and completely changed the ecology of the eastern forest. So it's not like it's not like what it used to be. Fusinia graminis, this is, causes black stem rust on wheat. What happens here is that the wheat, the fungus weakens the stem so that as it produces the grains, they fall over. And if they fall over, then they can't be harvested, and we lose all that. And so this is right now there's a type of, a strain of this that started in Africa that we don't have any way to control it. And it's, I think it's already moved into India, and it's sort of worrisome that it may make it into the United States, but right now it's not here yet. Ergot, this is during the Middle Ages. Instead of wheat, a lot of times they would make rye bread, and then there were many outbreaks of rye toxin because of something caused by the fungus. The fungus is called ergot, produces lysergic acid. This here, incidentally, is a beginning component of LSD. Okay, so this here led to people died from it, but also they saw a lot of things that weren't there. Okay, all right. So some of these fungi are mutualist. So I already talked about mycorrhizae, and there's two different types here of mycorrhizae: ectomycorrhizae, endomycorrhizae. So 95% of all vascular plants have these associations, and as I said, experiments have shown when you get rid of the fungus, then the plants, the vascular plants, do not grow very well. They're stunted. And so it is a mutualistic relationship. It's plus. Lichens. This is very interesting because that is not an individual organism. It's a composite organism consisting of usually an ascomycote fungus. A lot of times can sometimes be a basidiomycote fungus, and some sort of alga, okay? Usually a chlorophyte or cyanobacterium. And they're growing together. And so this allows, a lot of times you'll find these lichens in very dry conditions on rock, okay, for instance. And yet, usually the photosynthetic part, the cyanobacteria, the chlorophytes, they really require water. And so they're living inside of the protective covering of the fungus. And therefore, what the fungus is getting out of it, of course, is photosynthate, produced by these photosynthetic organisms. But what so that's what the fungus is getting out of it. But what is the chlorophytes getting out of it? They have protection, okay? And other mineral supplies that the fungus mobilizes. Interestingly, these can live apart from each other, okay? So they're not necessarily so dependent on each other. And when the fungus is by itself, the species of fungus that are involved here, its growth pattern is completely different than what it takes on in a lichen. And that's also true of the chlorophyte and cyanobacteria. They take on a different form when they're living by themselves. Okay, and this is just showing some lichens. Okay, this is sort of a foliose type of lichen, crust, and so forth. This is growing on a rock, okay, which is, and I already said that. Okay, and this is just some other pictures showing lichens, cross section, okay, through one. This is actually an ascomycote. The fungus itself is, but usually it will have a thick layer down here, sort of loose hyphae in between, and once again another thick layer up on top. And this is where the either green algae or cyanobacteria happen to be near the top here. Sometimes instead of these are reproductive structures. We don't need to go into how they do that. But sometimes they will produce a cup-shaped structure up here, which is the ascomycote, okay, the ascocarp. And that's all we need to say about fungi. So before I go on to animals, anyone need to see a slide here?
What's the difference between ecto and endomycorrhizae?
Okay, say what?
The difference between ecto and endomycorrhizae.
The mycorrhizae in general, that's an association between the fungus growing in the soil and the roots of plants. That there can be two different forms, ectomycorrhizae where the fungus's association with the roots is superficial, that is, it's not penetrating into the cells. It's still in contact with the plant root cells. And then endo, that's where they have special type of arbuscular hyphae which penetrate into the cells of the root cells. Okay? Yes?
Was the Pucina graminis the black stem rust? Was that the black fungus they were talking about last year? Like in India, and then it made
Yeah,
people lose hearing?
exactly.
Oh.
Yeah, yes, yes. Okay, all right, on to the animals. Our last big group to look at. Still, Amorphia is the clade, Pistaconta, and then Animalia. Okay, and we have a lot to say about animals because we are animals, right? So we're very interested in them. Okay, this is multicellular, of course, and the multicellularity here, although I'm not putting it up there as a synapomorphy, it's still convergent with multicellularity in other eukaryotes because it's independent. Of course, they're heterotrophic. In fact, this whole clade, Amorphia, it's primitively heterotrophic. That means that there's absolutely no evidence of any of their ancestors ever having had chloroplasts. And so they never underwent, none of the ancestors underwent any form of secondary endosymbiosis for obtaining chloroplasts. So now here the heterotrophic is still extracellular digestion, just like in the fungi. However, what the fungi do is that they grow through their food and they secrete digestive enzymes outside of their body. It's external, and then they absorb the digested products in. Animals do this too. However, they have a special, instead of secreting outside of their body, they have a special area inside the body, which is still technically connected to the outside. And it's where they ingest, take into this chamber food material and secrete digestive enzymes there. But technically, that is still outside of the body. Okay? Really, the food material that's broken down is not technically inside of the animal's body until it crosses into the bloodstream. Okay? And that's true with us. So we're donuts. All right? We have an opening at one end of our digestive system and opening at the other end, and that's all continuous with the outside. Okay? If you don't believe me, try this experiment. Get yourself some floss, swallow it, okay, one end of it, and wait for a few days, and then you can floss, all right? Okay. So, goes all the way through.
No, just looking onto your tube.
All right. So,
Onto your tube. Oh, God.
and here, carbohydrate reserves in animals stored in the liver and muscles, at least in vertebrates, as glycogen. They have no cell walls. Now notice I'm not mentioning any apomorphies up here yet. These are just characteristics. However, they do have, just in one part, in the eumetazoa, I'll get to the eumetazoa here in a moment, but a special type of tissue which will be an apomorphy for that clade, all right? But I'll get to it a little bit later here. And this is muscle and nervous tissue. This is something unique to this particular group of animals, which is the vast majority of animals. Vast majority of animals, right? And type of life cycle is diploid, okay? So diploid life cycle, the only haploid phase, of course, is just gametes. There are no spores in this group. It's all gametes. Now, these here are apomorphies for the Animalia. Okay, special types of protein junctions between cells. They're adhesion junctions. In vertebrates, these are called tight junctions. But here, I'm just going to make it a general statement because tight junctions are only in vertebrates. Still a type of protein junction that holds the plasma membranes of cells together. Okay, and furthermore, the extracellular matrix. Okay, ECM stands for extracellular matrix. What is the extracellular matrix? When I say extracellular matrix, what am I referring to?
Is it the outer complement boundary?
Okay, it's a loose network of proteins and structural carbohydrates that are on the outside of the cell. Okay, animals don't have cell walls, but they have extracellular matrix. And in there, they have this protein, okay, which is unique to animals, collagen. So extracellular matrix with collagen and various types of proteoglycans. Okay, so this is a complex proteins with structural carbohydrates that are covalently bound together, all right? So these two are apomorphies, synapomorphies for the animality. Okay, now, before I really get into any of the groups, there's something about the body plan. It's also called Kyle plan. You'll come across this word also, it means body plan, but that's from German. And about their structure, several different aspects of the structure that I want to talk about. One of these has to do with symmetry, okay? Plants are basically asymmetrical, all right? So their branches can grow any direction. But we do have some animals that are also asymmetric. They have no particular obvious plane of symmetry. Okay, when I say plane of symmetry, when you cut through the body, that it creates mirror images. Okay, a left side, right side. Some of the animals have radial symmetry. Okay, now what is radial symmetry? It means that through the long axis of the body, you can take several slices, okay, like cutting through a pie, that will produce mirror image right and left sides all the way around. Okay, so the good example here is like a tire on your car. That has where the hub happens to be, okay, that's the center part, but if you cut your tire in any direction, you come up with two halves that look the same, okay, mirror image. So they don't have really don't have a left or right, up or down. What they do have is an aboral side and an oral side. Okay, the oral side is the side where the mouth is. Okay, the oral side is the side where the mouth is, and the aboral side is the opposite side. Okay, and I'll show a picture here in a moment. Then we also have bilateral symmetry. The vast majority of animal groups are bilateral. Okay, it's a very, very big group. And here, there's only one plane of symmetry. You cut right down the middle, and you have a right side and a left side. So bilateral animals have an anterior part, usually that's where the head is. Posterior part has a left side, a right side, a dorsal side, which is the back, and a ventral side. Okay? But there's only one way to cut it in half and have two mirror images. Okay? And this is bilateral symmetry. All right? So, as I said, this has posterior, anterior. Anterior is where the head is. Posterior is the other end, opposite from where the head is. Left and right. Dorsal means back side. Ventral means belly side. Now, for bipedal critters such as ourselves, in human anatomy, these terms get very, very confused because we're standing straight up. And so sometimes this side is called the anterior side. When if you were the normal animal on all four, this would really be the ventral side. Okay, so some of these, for humans, some of these terms get confused. I prefer to look at the human body as being on all fours. So get on your all fours and we'll look at it that way. All right? Okay. So this here, Hydra, it's radial symmetry. Has oral side, aboral side, and it can be cut in many different planes. It's like the spokes of a bicycle, okay, around there. We have many planes of symmetry around a center axis. Whereas bilateral symmetry, there's just one plane through which you can cut the animal and have a right and left half. Okay, that's one thing, symmetry. So as we go through these groups, various clades, they're going to differ with regard to symmetry, okay, and as a defining part of the clade. But also there's another issue of what is called primary germ layers. All animals in the early embryology, right after fertilization, go through pretty much the same developmental stages, up to a later period when they deviate, depending on whatever clade it happens to be. So first of all, the zygote, fertilized egg, zygote, okay, it's diploid. When it begins to divide, it is a process that is called cleavage, and this is just mitosis. You undergo cleavage to form a hollow ball of cells, one cell layer thick. It's called a blastula. So it's one, and there's the cells all look the same. So this is just a hollow fluid-filled sphere that's one cell layer thick, and then the blastula will undergo what is called gastrulation. In which some of the cells, it continues to divide. Cells continue to divide, but some of the cells will start moving inward to form a second layer on the inside. And like up here. So there's the zygote, and when at 8-cell stage, we get to the blastula, and that hollow area inside is called a blastocele. So all animals will go through this, with the exception of sponges. With the exception of sponges. Okay? Blastocele, and then with gastrulation, so gastrulation is happening right here, is that some of these cells will start to move inside, compressing the gastrocele, forming one opening, and which is called a blastopore, and now we have two layers. Okay, our blastocele is still there. It's a fluid-filled space, but it's between this yellow layer and the blue layer. And of course, the real thing is not colored. This is just only for you to be able to tell them apart. And so this here, yeah, blastula. Go ahead and put the stuff up. So I'm going to go to the next slide. First of all, I'll let you get this down before I go to the next slide, because I think the next slide is a bigger presentation of this photo. Yes?
What is formed after the blastocele? Like, what's the...
It's the fluid fill. I'll show it here on the next slide.
Okay.
Okay. So I'll come back to this in a moment. All right? So this is easier to see. So if we cut through here, you'd have the blastocele on the inside. Okay? Once we get these cells moving in, our blastocele is becoming compressed, and we're forming another chamber. This chamber is the beginning of a digestive chamber, and it's called an arc enteron. Okay, arc enteron. So what we have here is called the gastrula. The gastrula. So the gastrula can consist of two primary germ layers, or there might be three. Okay, I'll go back and show you this in a moment. Right now, this here is showing just two layers. The yellow layer, which is inside, and a blue layer, which is on the outside. In between is a fluid-filled space called the blastocele. But within the yellow cells is a fluid-filled space too, which is the beginnings of the digestive chamber. This is called the arc enteron. And arc enteron. And the opening to that arc enteron is called a blastopore. Okay? Now, this outer blue layer, both of these two are primary germ layers. Primary germ layers. So let's go back. And if the group of animals at their embryology only up to this point, the gastrula, only contains two primary germ layers, it's referred to as diploblastic. Okay? So they have what is called ectoderm on the outside, endoderm on the inside. Now these two layers, they will form very specific parts of the body. Okay, so by this time cell differentiation has occurred, and they're destined to form very specific parts of the body. Other animal groups will have a third layer that forms in the gastrula, in between the endoderm, ectoderm, and this is called mesoderm. So if the early gastrula has 3 germ layers, then this is called triploblastic. Now, this diploblastic, triploblastic, especially the triploblastic, is phylogenetically significant. Okay, it is phylogenetically significant. Okay? All right, everyone have this. All right? Now, what the ectoderm will become, it will become the integument of the animal, the skin, the outer part. And in things like vertebrates, well, actually even more than vertebrates, those animals that have a well-developed nervous system, a lot of the nervous system originates from cells in the ectoderm, primary germ layer, right? The endoderm will become the digestive chamber, or the digestive tract, as the case may be, and associated types of glands. The mesoderm will become muscles and some other types of organs that are found in between the body wall and the digestive channel. Okay, so these are the primary germ layers. Okay, and here we're taking the embryology just up to gastrulation, okay, up to the gastrula. Beyond that, the development depends on the group of organs, the group of animals. Okay, they sort of differ as we go on. Another issue with the bowel plan, okay, the body form, is something called body cavity. What the body cavity happens to be, it's also called a coelom, and I'll show that word in a moment. It's a fluid-filled space other than the digestive tract. Now, eventually, that blastocele disappears. Okay, the blastocele disappears, all right? So we're talking about something else. So this is body cavity, develops later in the embryo, and it's a fluid-filled space that's found between the parts of the body that came from ectoderm and the parts of the body that came from endoderm, and usually it is within the mesoderm, very much. And so this is only in the group of bilateral animals. So body cavity is only an issue with triploblastic animals, bilateral animals. If they're radial animals, you don't talk about body cavity. And if it's diploblastic, you don't talk about the body cavity. Body cavities are only found in the bilateral triploblastic animals. And there's 3 different variations, okay? Some of them don't have a body cavity. And used to be that it was thought that this was the plesiomorphic state. We now know that it is not. It is that they've lost the body cavity, okay? And we'll come back to that here in a moment. So they're triploblastic, but there's no fluid-filled space. And so if you were a pin and you're poking it through the body, the only space would be the lumen of the gut, the lumen of the digestive tract. So first of all, you'd be putting that pin through tissues of ectodermal origin, right into tissues of mesodermal origin, right into tissues of endodermal origin, and then into the digestive lumen. Okay, no fluid-filled space. Coelomates, okay, so coelom is this word. Just change um at the end and you have coelom. That always means a fluid-filled space, coelom. So this body cavity is also called a coelom, right? So coelomates, they have body cavity that is within the mesoderm, And so if you poked a pin through them, you would, first of all, the pin would go through tissues of ectodermal origin, into mesoderm, into the body cavity, fluid-filled space, into mesoderm again, into endoderm, and then into the gut. And I'll show pictures. What's that? Well, I'll go over it again. All right, just a minute. And then there's one other type that's called pseudocoelom. They do have a fluid-filled body cavity, however, it's only lined with mesodermal tissue on one side instead of both sides. And I'll show that here. And so that's called a pseudocoelom. So this here, an acoelomate. We call them acoelomates, like a flatworm. There is no body cavity. So the blue is ectodermal tissue. The red is mesodermal tissue. And the yellow on the inside, that is endodermal tissue. And so there's no fluid-filled space. There's a cavity for the gut, that's all. But that is not what the coelom happens to be. This is a little bit complicated because a lot of times in the mesoderm we have internal organs. But here you'd be going through, first of all, blue is ectoderm. The red is endoderm. This green, or not endoderm, mesoderm. Red is mesoderm, and the green is a fluid-filled space. It's the body cavity. And notice here that that body cavity, fluid-filled space, is surrounded on all sides by red. So it's completely within the mesoderm. And then going from the mesoderm, you would go into the intestine, which would be endoderm. So here, for simplicity, if you were putting a pin through a coelomate, you'd first of all be crossing through ectodermal tissue, mesodermal tissue, body cavity, fluid-filled space, mesoderm again, endoderm, and then into the gut. Now, what is the significance of coelomate? Well, I'll come to it here in a moment. Nematodes, pseudocoelom. They do have a body cavity, but it is not completely surrounded by mesodermal tissue. So here you would be putting a pin into it. You'd be going through the ectoderm, mesoderm, body cavity, the pseudocoelom, and then endoderm, and then the gut. Okay, so the body cavity is only surrounded on one side, the outside, by tissues of mesodermal origin. Okay, that's pseudocoelom. All right. Now, what's the significance of this space? One thing, Cushing's organs. Okay, Cushing's organs, and also that fluid-filled space allows animals to get larger. Okay, and increase the number of organs, internal organs. And furthermore, now the internal organs can move independently of the body wall. Now, if we didn't have a body cavity, every time your intestine or stomach happened to move, you'd see all this wiggling on the surface of your body. Okay, that would come through. And furthermore, that movement of the digestive system would be impeded by the immobility of your body wall. There would be competition there. Furthermore, anytime you bent over or deformed your body wall, it would affect your digestive system and internal organs. That having that fluid-filled space there allows these organs to not interfere with each other. Okay? And furthermore, in some animals, such as earthworms, they have no bones, but yet that fluid-filled body cavity provides a place of resistance upon which skeletal muscle can contract and allow them to move. And so it serves as what's called a hydrostatic skeleton. Okay? And so that's some other functions here of the body coelom. Okay, so now for the phylogeny. Where did all the erasers go? Where?
On the whiteboard.
Right next to the.
Oh, there. this. Here we have Animalia. This is also called metazoa. Okay, they're equivalent. We're going to be using Animalia here, but in your future studies, you will come across that word metazoa. Okay, so that's the same thing. So I've said here that we have extracellular matrix with collagen junctions, protein junctions. I'm just going to abbreviate it here. All right, so one of the first groups here is called the peripherum, and it's a basal group. They have no tissues or organs. They're multicellular. They are multicellular, but they have no tissues or organs. And they're asymmetrical. Asymmetrical, but as an apomorphic for this, what they have is this unique water system for circulating water to get their food. And it's called a internal aquiferous system. I'm going to put up here, internal aquiferous system. That's all I'll give, and I'll show that here in a moment. So here with the body, these are sort of base shaped quite a bit, you know, but yet the way in which chambers inside of the body happen to be arranged, they're asymmetrical. However, there's an internal fluid-filled space that's continuous with the outside. It's called the spongial seal. So you see that suffix, C-O-E-L, it always means a fluid-filled space. Okay, this is not the same thing as a sebum. Okay, not the same thing as a sebum. They have different types of cells. Some of these have flagella. These are called choanocytes, or also collar cells, because, well, I'll show pictures of them in a minute. But these are the cells that digest food, okay? In this group, the periphery, they do not ingest their food. So the digestion is not extracellular, like in the rest of the animals. This is an exceptional group. They digest inside lysosomes and the choanocytes, and usually this is very small particles, bacteria, the eggs of other animals and so forth, sperm of other animals, and then that digested material is spread by amoeboid types of cells that look like amoebae, that move throughout the body and distribute the food to other cells. They really don't have a digestive system except for internally. They still have skeletal material, because some of these sponges can still get pretty big. And even though most of them are marine, there's a few freshwater sponges, they still, animals have no cell walls, unlike plants, and therefore not a whole lot of support. So they have these things inside called spicules, and I'm putting that as an apomorphic too. That's their skeletal system. And depending on what those spicules are made out of, sometimes it's silica, in which we call them glass sponges. Some of them are chalk, calcium carbonate, and they're called chalk sponges. And then some of it isn't flexible organic material, usually a type of protein. And in fact, now the sponges that you use in your kitchen, those are artificial sponges. They're not real sponges, okay? They're not made up, don't come from these animals. However, sponges used to, okay? And they were all of this group, all right? And in the lab tomorrow, you will see is I'll have just only the organic sponge. In fact, if you go to a car wash where they wash your car, and they're using real sponges, okay? And these come from the skeletal material, flexible skeletal material of this particular group of sponges. All right, now, the body has only two layers. It's an epidermis on the outside and something called a gastrodermis on the inside, okay? In between those two layers is an extracellular matrix that's called mesothelium, Okay, I'll show pictures here in a moment. Yeah, just look at a few of these. First of all, make sure everyone has all that. We're not going to go, so these do consist of several cell types. Now, why do we say they're not a tissue? Because you can take a sponge, body of a sponge, and squeeze it through a sieve, completely separate all of the cells, and put them out on the table here, and put some of them way over there, and some of them way over there, and the cells will just sort of come back together, okay, and form a sponge independently. And so you didn't kill it by doing it that way. With a tissue, you have cell interdependence. You can't separate the cells, and it'll keep living.
You mean like the separate parts will form new sponges, right?
Yeah. So you can take all the cells in the body of the sponge, separate them out, and they'll crawl back together. Yeah.
So once you separate them, will come back together, or will they?
Yeah, no, you separate them all, and they will eventually come back together, yes.
How do they find each other?
Yeah.
Chemical signals. Okay, you know, always there's some types of chemical signals that are used. Okay, so this is a type of sponge. Now you might say, well, isn't that radial symmetry? Well, this whole thing is a sponge, one individual. That's not radial symmetry. Might have a little piece here. Even if you cut through this, you'd find that all the pores and inside here there'll be other types of chambers. They are not. They're asymmetrical. Okay, the way they're put together. And this is just one sponge species. This is just one individual. This is not 1, 2, 3, 4, 5. This whole thing is just one sponge. Okay, so this is a simple body like this. On the outside, epidermis. In fact, this is better. Epidermis, and then this here is called gastrodermis. The word isn't up there, but it's a layer of color cells. In the middle is this chamber. That's the spongial seal. That's the spongial seal. Now, what is the internal aquiferous system? The beating of these flagella on the collar cells creates a water current. And all through the body wall, there are these pores that are found, that are formed by a particular special type of pore cell. And so as these flagella happen to be beating, it brings in water from the outside through these pores, and they have thousands and thousands of pores all over their body, and into the spongial seal, circulating up and out what is called, the opening at the top is called the osculum. Okay, so this here, of course, is continuous with the outside world. But as they're bringing in this water from the ocean, it has bacteria, it has other organic stuff that is in it, and they get caught on this sort of sticky collar that's on the surrounding the flagella. And this is where the food particles are then brought into the, by endocytosis, into the collar cells, where it forms a food vacuole that then binds to a lysosome, and the material gets digested. And then there's amoeboid cells that are moving, come into contact with these collar cells, pick up some of this food, digested food material, and spread it throughout the body. So this right here is the mesohele. That's not cellular. There may be amoeboid cells crawling through there, but this is extracellular matrix.
So they take in the water and they absorb all the particles.
Yeah, yeah, exactly. And they keep circulating this water through their body. And that's how they digest their food, okay? And so they're strange. And I'll say nothing more about the peripheral, okay? There's some freshwater groups, but we've got to move on, all right? Because we have a lot of animals to go through. Peripheral is a sister clade to Eumetazoa, not metazoa. Metazoa includes the sponges, the peripheral. Okay, so this here is the Eumetazoa. Eu meaning true. Okay, these are true atoms because they have true tissues. Okay. In other words, you have a tissue. A tissue is a collection of cells that two or more different types of cells that are interdependent, okay? You can't separate cells apart from each other, and they work together. So this has body symmetry. The periphery are asymmetrical. Okay, body symmetry. And they have at least tissues, and usually organs as well. All right? Now, the periphery, their embryology is not like what I was talking about before. Okay, starting with a zygote forming a blastula, forming a two-layered gastrula. That doesn't happen with periphery. Okay, doesn't happen with the periphery. It does happen here. So we do have here then true ectoderm. Although the sponges have two layers, and even in their embryology they have two layers, they're not characterized as primary germ layers. It's one group that doesn't have it. Epithelial tissue. Okay? Epithelial tissue. These epithelial tissue and epithelium, and we'll have, when we get into the animal physiology, we'll have a lot to say about epithelium. These are lines, body cavities, the outside of the body, and many of the organs are based on epithelium. This is a special type of tissue, where cells are held together, the plasma membranes are held together by those protein junctions. Okay, those protein junctions, which in vertebrates are called tight junctions. Invertebrates. And we'll come back to that a little bit later. And they have nervous tissue. For some of these groups, it's rather, let's just say nerve cells. Cells that happen to be able to conduct electrical signals. So there's a lot of apomorphies here. Now, out of the eumetazoa, two branches. One, radiata. The other branch, bilateral. Two branches here. So, although it's hard, this is probably a plesiomorphy. Nonetheless, I'm going to count it as an apomorphy. Is that the radiata have radial symmetry? Whereas bilateria has bilateral symmetry. Furthermore, these are diploblastic. Okay. Now, I'm not counting that as an apomorphy. This is an apomorphy. Okay. Now, it's probably really a plesiomorphy, but just so that we have something to hold on to, because the earliest animals were that we have in the fossil record happen to be radially symmetrical. But if we were using outgroup comparison, we would be have to use the periphery as an outgroup to polarize which of the two character states, radial or bilateral symmetry, is plesiomorphic or apomorphic. Well, we don't have that type of symmetry at all in periphery, so we can't polarize. All right, so these only have two primary germ layers, bilateria, bilateral symmetry, and they are triploblastic. Since I put ectoderm, endoderm as an apomorphy for eumetazoa, to make bilateria then triploblastic, it means mesoderm is the apomorphy. Okay, mesoderm. They have three germ layers. Now, within the radiata, we're only going to look at just one group, and that's Nidaria. However, I will let you know that there's others. Other groups there, okay? So just like with Rhizaria, foraminifera, Rhizaria are different, okay? They're different clades. One is within the other. And that's true here with Radiata. We're only going to look at Nidaria. These are hydras, jellyfish, sea anemones, and corals. I have live hydras for you to be able to feed tomorrow. Okay, you can watch them eat. Okay.
Is body cavity considered synapomorph or anapomorph, or is that part of mesosome?
That's going to be up in the bilateria. Yeah. So this, they don't have a body cavity. So anything that is diploblastic, the issue of a body cavity isn't there. It's only in the bilateria that we have body cavities. And I'll get to that in a moment. You don't need to know this word. This is the name of the clade for what are called comb jellies. Yes?
So true tissues and organs are an apomorph?
So what?
True tissues and organs are an apomorph?
Uh,
On the first one, is that an apomorph, the true tissues and organs?
At the very
Possess
top,
true
the very
tissue.
first.
up here?
First line.
Yeah.
No, these, only where I have the stars.
So then you wrote tissue on the.
Yeah, you can put tissues over there. It's sort of redundant because that's nervous, that's tissue. Yeah, that's redundant, so let me leave that out because nervous tissue, epithelial tissue, that's a tissue. Okay, so no point in putting tissues up here. Because with those other two groups, we have tissues. Okay, so hydras jelly, so let's look at Nidaria, because you'll be looking at them. This is a comb jelly, and we're not going to consider them, okay? So, but know that they're there. You don't need to know the word Ctenophora, just only comb jellies that they exist. And this is in the Linnaean system, Nidaria is treated as a phylum. Now, once again, I'm not going to pay attention to Nidarian, or excuse me, Linnaean classification, still we'll put it up there. So this is hydras, jellyfish, sea anemones. They have radial symmetry. I already have that as an apomorphy for Radiata. And they're diploblastic. I already have that as an apomorphy for Eumetazoa. Okay, and so that's true for everyone above that. They do, between their endoderm and ectoderm, they do have an extracellular matrix that's sort of jelly-like. Okay, it's called mesoglia. However, in its composition, it isn't the same as mesohyal that we have in sponges. But nonetheless, it's sort of similar in many ways. Okay? The only organs that they have. Now, what is an organ? An organ is a structure that's made out of two or more tissues. Okay? Two or more tissues that work together for a physiological function. And so for this group, that's the only organs they have. All right? Otherwise, they're just at the tissue level of development, and but the gonads, testes, and ovaries. Okay? Their digestive system, from here on, we now have extracellular digestion. Okay? There's some sort of digestive chamber for all of the Eumetazoa. Okay? I'm not putting it up as an apomorphic because you have plenty of that now, but there's a digestive chamber. In this case, for the Nidaria, this is called a gastrovascular cavity. This is in contrast to a digestive tract. A digestive tract has two openings to it, a mouth and an anus. And food flow in a digestive tract goes in one direction. For a gastrovascular cavity, there is only one opening that serves both as mouth and anus. And so the gastrovascular cavity is a bag, okay? Things get digested there, but no specialization along its length. So they have, and these are some things that you'll be looking at tomorrow. These are hydrozoans. Okay, that's a group up there, hydrozoans, scyphozoans, sea anemones, and corals. All right? But they have radial symmetry. Radial symmetry, oral side, and they have oral side. nervous system, there's no brain. Okay, there is no central brain there, but they still control their muscles. They can still move, and that is arranged in what's called a simple nerve net. So there's no nerves. These are just neurons that are interconnected with each other very loosely, but they can still control the movements of the body. This is an apomorphy I'll put for the cnidaria. Cnidocytes. These are special steaming cells. And what they have is a special organelle. So the cell is called the cnidocyte. So that's the apomorphy, that type of cell. This is an organelle. Okay, that's a special type of organelle, the nematocyst. And how many of you have ever been to the ocean, wading around, and have become stung by a jellyfish?
Oh, so that's what that is?
That's what you're getting stung by, okay? So when you get stung by a jellyfish, it is due to these cnidocytes. These are usually concentrated around the mouth and on tentacles. So I'll come back to that here in a moment. This is the hydra. And you'll have some hydras tomorrow, and I'm going to give you probably Daphnia, so that you can have your jollies watching the hydra eat a Daphnia. Okay, that's a water flea. All right, so the mouth is right here, have tentacles. This is where they catch their food and entangle it and bring it into a gastrovascular cavity. So there's just one opening, brings it in there. And this is a cnidocyte. So it's covering around the mouth and on the tentacles. You'll see the cnidocyte. Usually there's a modified cilium, which is a trigger, and this is the nematocyst, okay? That's a special type of organelle that is put into here. And when this is stimulated, water pressure changes quite a bit and pushes out the tangled nematocyst, and they can be quite long. And so this can coil around their prey, but also they will secrete toxins. So that a lot of times they're secreting a toxin which can immobilize their prey, and then they bring it into the mouth for digestion. Okay, so that's how these things work. All right, now in their life cycle, there's two different body forms. Something called a polyp, and these alternate with each other. A polyp is, when I say sessile, it means it's attached to a substrate, it doesn't move around. Oh, it can move back and forth, it can move their tentacles, but it doesn't move from one place to another. So it's attached, and the mouth is up, okay? The mouth is up, and the mesoglea is very, very thin. Okay, so that hydra that I showed, back here, if I back up, actually let it go forward. This happens to be a polyp. But then there's also a form called a medusa. And the medusa and polyps, they differ in this way. The medusa, primitively, ancestrally within the group, it's free swimming. It's the jellyfish. Okay, that's a jellyfish. Okay, free swimming. The mouth, when it's moving, it is oriented downward instead of upward. And the mesoglea is thick. There's a lot of mesoglea, all right? That's what makes them jellyfish. So, typical life cycle, but let's just go back here for one moment. Now, within this group, Cnidaria, there's 3 different subgroups, and I'm going to put them up here, but I don't think, in order to speed up getting through the animals, I don't expect you to know these names, okay? In fact, on a phylogeny, I will never have you do 3 different branches here. But still, bear in mind that there's 3 major groups here, something called hydrozoa. These are basically the hydras. And in their life cycle, they alternate between a polyp and a medusa. Whereas in the other 2 groups, most of the things that we call jellyfish, we have medusae in this group, but most of the things you see as jellyfish happen to belong to this group. And they have mostly lost the polyps. They don't have polyps. They're only medusae. These are the more plesiomorphic ones. Whereas the anthozoans, they're the other way around. These are corals and sea anemones. They don't have the medusa. They're only the polyp, and the polyp is sexually reproductive. So in their life cycles, and this is an anthozoan, is that they grow in colonies. Some of these are sexual polyps. This is one individual, one individual, one individual, one individual, but their gastrovascular cavity, they share it. Okay, they all share gastrovascular cavity. So they're sort of strange looking things. And some of them are reproductive. So by mitosis, it will produce a plug here that will bud off and become a medusa. The medusae have testes and ovaries. Okay, they're in the hydrozoans. These are reproductive, separate female and males. They produce sperm and egg, which they release into the water, and fertilization takes place. Zygote goes through embryology, and that becomes what is called a larva, and then that larva will eventually settle down on a substrate and grow into a polyp, and the polyp, by mitosis, keeps on growing, producing other polyps. Yes.
Is it one of the hydrozoans that share a body cavity?
I can't, let me see here. No, I think corals do it too. Yeah, corals will do it too. So, but not sea anemones. Okay, yeah. But hydrozoans, this is very, very common. They have a colony, then produce medusae. These are real small. They usually don't get very big. Unlike the scyphozoans, these are big jellyfish. Okay, so this is a jellyfish, scyphozoan, that belongs to those groups. They don't have polyps. Okay, and this is another type of jellyfish, scyphozoans. And the anthozoans, these are only polyps, no medusae. And the polyps have, in their evolution, basically the medusa has been retained within the polyp because these guys have gonads. Okay, these guys have gonads. So corals, these are polyps. And sea anemone, right there, they can move a little bit, okay, but they're not very graceful, not very fast, all right? But they're frequently eaten by starfish. Okay, and that's all I'm going to say about Cnidaria. Going on now to the next clade, Bilateria. This is the biggest group. And this, I already put up there, bilateral symmetry. Mesoderm, they're all triple belastic. Yes?
So why is each like branch considered a different one if it grows from the same zygote?
That's a good, curious thing. That's probably our calling individuals and things like that. What is an individual? We're sort of having a bias of our own structure. Because it is true. We see this polyp, it is a unit, but yet we have them together as a colony, but it's sort of, that was produced asexually. And of course, their gastrovascular cavities are together. Is this whole thing colony should be called that individual, should be called this? So it's a real individual. Okay, now, one of the significance of mesoderm is this tissue, muscle. Okay, it's one of the derivatives of mesoderm. And so this group has muscle. Since I put up here mesoderm, I'm not putting muscle up here as an apomorphism. All right? And some degree of cephalization. Now, what that word means, cephalization, is that there's some sort of head. Because as these bilateral animals are moving into a new environment, that is the best place to concentrate sensory organs, to know what you're getting into. You know, you're going to run into food, you're going to run into a mate, or you're going to run into something that's going to eat you. Okay, so this is sort of the head. In other words, Since these things are moving in a directed motion, then there's one part of the body that hits the environment first, and that is a good place to put a head. Concentrate sensory organs. Now, of course, jellyfish still move, but they sort of face their environment from all directions. Even though they do move, they're still facing their environment from all directions. Okay. Now, in some groups, of course, cephalization is very intensive, like humans, animals, vertebrates, insects. They have very clear heads. Body cavity. Okay. I'm going to say that this is an apomorphic body cavity. Some groups have lost it, okay, the body cavity. And hence are acoelomate. So we have coelomates, those are the ones that have true body cavities. The fluid-filled space is completely within the mesoderm. Pseudocoelomates, we believe that that is derived from coelomates. That simply what we have is that they had coelomate ancestors, but they have developed the loss of the mesoderm on one side of the body cavity. Okay? And then a few of them independently, several groups, have become acoelomates. It used to be thought that these were the most primitive, that acoelomate evolved into pseudocoelomates, coelomate into coelomates, but with molecular data, the interpretation is much, much different, because the molecular data really supports some of these being real high in the bilateral tree, and therefore it appears that they have secondarily lost the body cavity. Okay? Now, here, once again, two major branches. Protostomia. And the deuterostomia. Protostomia, deuterostomia. And this is just only from your textbook showing this protostomia right here, bilateral, deuterostomia, protostomia. Then the protostomia is divided into two other branches, Lophotropozoa and Ecdysozoa, and then we are up here in the deuterostomia. Okay, so I'm going to separate these with our lab exercises. Exercise 8 is looking at porifera cnidaria, and exercise 9 is going into protostomia, Lophotropozoa, that part. Okay? I'm sort of running out of time here. I will come back to this. Okay, you can still read about it, but right now I want to just quickly go through some of the groups that you'll be looking at tomorrow. Okay, so I can just be ahead. Then I'll come back on Tuesday and cover this. So let's just get through here. I'll talk about this a little bit later. Because I just want to introduce you to the groups that you'll be looking at. Is we're going in a branch called, for exercise 9. Lophotropozoa. Okay, then exercise 10. Ecdysozoa. But this is for exercises that we're getting into tomorrow. So within here, this is a Linnaean phylum, Platyhelminthes. So put it up here. And I'm just going to go through it real quickly. I will go over this again, but right now I'm not going to concentrate, so on Thursday I'll go over this. Let me just go through the groups. They're acoelomate, okay? So that's an apomorphy. They've lost the coelom. And they have a gastrovascular cavity, so not a complete digestive system. One opening serves both as mouth and anus. Simple nerve net. However, they do have nerve cords, okay? Although most of the nervous system, they do have some cephalization. There are ganglia in the head that serves as a brain, but they have ventral nerve cords running down the bottom, okay? Some degree of cephalization, and as I said, I'm going to go through this real quickly just to show you some of the groups. And I'll cover all this again on Thursday. So dorsal ventrally flat, that's why they're called flatworms. And so you'll be looking at some of these tomorrow. So I'm not even going to talk about the apomorphies right now. I just want to show you the groups that you'll be dealing with tomorrow. Okay, so we'll go over here, but one of the things I have is free living planaria, and I believe that they have come in, and you'll get to play with them tomorrow. And then there's tapeworms. You'll get to look at some tapeworms, and I'll have stories to tell you about tapeworms on Thursday. I have a lot of tapeworm stories.
That's concerning.
And trematodes. These two are parasitic groups. So these are called flukes, and there's a lot of stories I can tell about them. Tubularians, they're planaria, they're free living. So these are the groups of the tapeworms. Another group you're looking at is mollusca. Okay, mollusks. And it's a big group. And I'm just going to go through some of the subgroups here. We'll talk about their characters later. You don't have to know this word, Polyplecophora, just call them chitons. I just show them that they exist. We're not going to talk much about them. But gastropoda, snails. Okay, these are snails and slugs. Bivalvia, these are clams, oysters, and scallops. These in the Linnaean system are classes. Cephalopoda, squid, octopus, nautilus. Okay, and these are some of these have evolved pretty good intelligence, very intelligent group. And then the third Ophitrochozoan is analytic. Once again, I'm not going to think this is tritone. Annelids, this is segmented worms. And there's, once again, 3 groups here. We'll talk about the characters a little bit later. Polychaetes, these are called feather worms. I'll show you pictures. Clitellata, this is both earthworms, Oligochaeta, those are earthworms, and Hierodenia, these are leeches. I have bought about 3 nice leeches, about that long. You guys can play with them tomorrow.
I don't wanna play with
Yeah,
them,
you can
but-
feed them if you want. Yeah, you can.
I can feed it?
I'll let you. Yeah.
Feed it.
They're nice and clean. They don't transfer any diseases. I don't think these are medical leeches.
Oh.
And so they could, but it takes a long time. And I'll make you wash your
each one,
hands
right?
very thoroughly
Because,
before you ever
you know.
touch the leeches, because oils and salts on your skin, very irritating. But I have light leeches. I have light
I
leeches.
love you.
And so just look at a few pictures, then I quit.
Be my guest.
Okay?
We're gonna go over this again.
Oh, yeah, I'll go all the way back. Okay. Right now, I just wanted to get over just enough to see, so you know what the groups are in exercise 9.
All right.
Okay. Exercise 9 is Lophotrochozoa. Okay. Platyhelminths, annelids, mollusks. We won't do the dissection of the earth.
Okay.
Okay, that I'm putting back, all right? So this is a leech, that's a finger, okay? But these guys have suction cups, and so, yeah, they don't do that. Earthworm and polychaete. And so those are the things you'll be looking at tomorrow. And then for exercise 10, which you do on Monday, 2 groups here, exozoans, the nematodes, and the arthropods. And that's where I'll quit. But yes, I'm going back. I'll go back on Thursday. All the way to here. I'll start on Thursday here, 25. Okay? All right.